In the year 2175, humanity embarked on a transformative journey that fused the frontiers of space exploration with the boundless potential of synthetic biology. On a crisp morning at the sprawling orbital research complex above Earth, the Genesis habitat, an unprecedented synthesis of biology and engineering, was about to begin its voyage beyond the known confines of the solar system. The pioneering scientists and engineers of this new era believed that the key to long-term space colonization lay not in conventional spacecraft design, but in the very fabric of life itself. Their ambition was to create self-sustaining ecosystems, engineered organisms, and adaptive biostructures that could thrive in the harsh environment of interstellar space, ensuring that human civilization could flourish on distant worlds. The Genesis project was conceived as a response to Earth's dwindling resources and the ever-growing demands of an expanding human population. At its heart was synthetic biology, a field that had evolved over previous centuries into a sophisticated blend of genetic engineering, bioinformatics, and nanotechnology. Scientists had learned to design and fabricate biological systems from scratch, tailoring organisms to perform specific functions such as energy production, waste recycling, and even the construction of habitat structures. The Genesis habitat was a living spacecraft, its walls pulsating with bioengineered cells capable of self-repair, its life support systems driven by colonies of synthetic microbes that converted cosmic radiation into usable energy, and its ecosystem designed to mimic the delicate balance of Earth's biosphere. In a grand ceremonial launch attended by leaders, scientists, and citizens from around the globe, the project's vision was unveiled as much a story of hope as a bold technical challenge, harnessing the unpredictable beauty of life to conquer the cold, dark void of space. As the countdown began, the architects of Genesis recalled ancient myths of Prometheus and Icarus, drawing inspiration from humanity's timeless desire to push boundaries while grappling with the inherent. This mission was not merely a voyage through space, but a leap into a future where life itself would be reimagined, reborn, and propelled into the cosmos by human ingenuity. On board the Genesis habitat, every component was a marvel of biological design. The outer hull was not forged from metal or inert composites, but was grown in specialized bioreactors, layer upon layer of genetically modified proteins and polymers, forming a resilient yet flexible barrier against micrometeoroids and cosmic radiation. Inside, luminous bioluminescent panels provided a soft ambient light mimicking natural daylight, their glow regulated by circadian rhythms programmed into living cells. The spacecraft's interior was an intricate tapestry of organic corridors, bioengineered gardens, and integrated laboratories, all coexisting in a state of symbiotic harmony. This vessel was not only a means of travel but a living, breathing extension of humanity. As the launch sequence commenced, technicians monitored streams of data from biosensors embedded in every tissue of the spacecraft. Each reading was a note in the symphony of life that constituted the Genesis habitat. The excitement was palpable. This was the culmination of decades of relentless research, countless experiments, and the merging of art with science. With a final surge of energy, the Genesis habitat was set into motion. Its synthetic heart pulsed with promise as it began its voyage toward the stars. Before the dawn of the 22nd century, synthetic biology had emerged as a revolutionary field that reshaped medicine, agriculture, and energy production on Earth. Breakthroughs in gene editing and the development of programmable organisms had given humanity unprecedented control over the very building blocks of life. In the decades leading up to 2017-5, scientists from every corner of the globe collaborated to create biological machines, living systems that could sense, compute, and respond to their environment. These early experiments laid the foundation for the Genesis project, as researchers discovered that living cells could be engineered to perform tasks no conventional mechanical system could ever replicate. In laboratories scattered across continents, biologists meticulously crafted microbial consortia capable of converting solar energy into chemical fuel, degrading toxic compounds, and even repairing damaged tissues. The iterative process of design, build, and test in synthetic biology became akin to writing the very code of life. Every success fueled dreams of harnessing these capabilities for space exploration. The vision was clear: to build a spacecraft that was not merely a metal vessel, but an extension of nature itself, a self-healing, self-regulating organism that could adapt to the extreme conditions of space. The promise of synthetic biology in the cosmic arena was multifaceted. Engineers envisioned reducing spacecraft mass by growing components in situ, rather than launching heavy conventional materials from Earth. Environmental scientists saw the possibility of creating closed-loop ecosystems where every waste product was recycled by engineered microbes, transforming byproducts into valuable resources. Even the tantalizing prospect of terraforming distant planets emerged, with synthetic organisms designed to alter atmospheric compositions and generate fertile soils. As debates unfolded in academic halls and policy chambers, consensus grew that the future of space exploration would be written in the language of DNA, and that the fusion of biology with technology would unlock entirely new horizons for human civilization. The Genesis project was conceived not merely as an engineering marvel, but as a philosophical statement. Its architects believed that by embracing synthetic biology, humanity could overcome its terrestrial limitations and pen a new chapter in its history. A chapter defined by resilience, creativity, and the seamless integration of living systems into the fabric of space travel. This bold vision set the stage for an extraordinary journey, one that promised to redefine the very essence of life among the stars. The construction of the Genesis habitat defied traditional engineering by blurring the lines between biology and technology. Rather than assembling prefabricated parts in sterile factories, scientists and bioengineers cultivated the spacecraft's structure using advanced bioprinting techniques and genetically modified organisms. Enormous bioreactors, cylindrical vats filled with nutrient-rich solutions, provided the nurturing environment in which layers of biopolymers and protein matrices were grown. Over many painstaking months, the living structure took shape, its organic tissues interwoven with nanofibers and embedded with sensors that ceaselessly monitored its integrity. At the heart of this construction process was a breakthrough known as biological self-assembly. Drawing inspiration from the natural processes of embryonic development, engineers programmed cells to follow precise instructions, organizing themselves into complex three-dimensional forms. The result was a spacecraft whose architecture was both functional and artistically organic, a graceful blend of science and nature. The Genesis habitat's walls were alive, capable of repairing microscopic cracks and adapting dynamically to fluctuations in temperature and radiation. Inside, corridors lined with engineered luminescent cells guided the crew through living chambers that pulsated with a steady, almost human heartbeat. Every structural component was designed to work in harmony with the integrated life support systems. Engineered algae and photosynthetic microbes were interspersed within the walls, contributing to oxygen generation and carbon dioxide absorption. Nutrient channels reminiscent of vascular networks ran along the interior, circulating essential compounds to critical areas. The fabrication process was a delicate balance between precise algorithmic control and the inherent spontaneity of biological growth. The end result was a spacecraft built not only for the rigors of space travel, but also as a living testament to the transformative potential of synthetic biology, a creation that embodied the union of life and machine. As the Genesis habitat neared completion, its leadership unveiled a comprehensive set of mission objectives that reached far beyond simple transportation. Central to the mission was the creation of a sustainable, closed-loop ecosystem capable of supporting human life for extended periods in the unforgiving vacuum of space. This ecosystem was not a static collection of parts but a dynamic, evolving network of synthetic organisms, engineered plants, and carefully balanced microbial communities, each designed for a specific function. The primary objective was to ensure that every byproduct of human activity was recycled, repurposed, and reintegrated seamlessly into the living system of the spacecraft. To achieve this, scientists developed a series of bioengineered symbionts that could coexist harmoniously with the human occupants. These microbes were tailored to break down waste products into useful nutrients while generating energy and sustaining critical biochemical cycles. Engineered for resilience, these symbionts could withstand the harsh conditions of space, from intense radiation to the challenges of microgravity. Embedded within the vessel's walls, these organisms formed a biological network that continuously monitored and regulated air quality, water purity, and nutrient levels. Specialized algae, cultivated within transparent bioglass panels, harnessed available light to produce oxygen, while their growth rates were modulated to meet the dynamic metabolic demands of the habitat's denizens. Genetically optimized plants played an equally vital role. Modified for rapid growth and enhanced nutritional value, these flora were arranged in vertical gardens spiraling along the interior corridors. Their roots intertwined with synthetic scaffolding to form living lattices that not only provided fresh food but also assisted in waste recycling and water purification. Advanced sensors dispersed throughout the ecosystem transmitted real-time data to an onboard AI, which adjusted growth conditions to maintain equilibrium within the closed-loop system. The interplay between these biological systems was a delicate dance of interdependence. Each organism, regardless of size, contributed to sustaining life aboard the Genesis habitat. Beyond sustaining life, the mission's objectives embraced a broader scientific ambition. The Genesis project functioned as a living laboratory, a platform for real-time study of synthetic ecosystems and their adaptation to the extreme stresses of space. Every day aboard the vessel presented new opportunities to observe evolutionary processes as engineered organisms interacted, mutated, and evolved in ways that defied conventional biological paradigms. The insights gained promised not only to revolutionize space exploration but also to deepen our understanding of life itself. The day of launch arrived amid an atmosphere of hushed anticipation and exuberant hope throughout the orbital research complex. In a meticulously choreographed ceremony, scientists, dignitaries, and citizens worldwide gathered to witness the historic moment when the Genesis habitat would leave Earth's nurturing cradle. The vessel, its bioluminescent surfaces shimmering like living opals, was gently detached from its orbital berth by a fleet of robotic shuttles engineered specifically to handle a living structure. Every step of the separation was executed with exacting precision, for even the slightest perturbation could disrupt the delicate balance of the habitat's bioengineered systems. As the final countdown resonated through the control center, the launch sequence initiated a series of synchronized events. The propulsion module, a marvel that merged advanced plasma drives with bioengineered energy converters, activated with a quiet hum rather than a thunderous roar. Unlike traditional rockets, the Genesis habitat ascended with measured grace, a smooth departure that reflected its organic nature. The propulsion system was calibrated to deliver gentle, continuous acceleration, allowing the onboard ecosystem to gradually acclimate to the stresses of launch and the transition into microgravity. In those first precious moments after liftoff, sensors embedded in the vessel's living hull recorded subtle fluctuations in cellular activity, as the structure adjusted to the absence of Earth's gravitational pull. Within hours of separating from its orbital cradle, the Genesis habitat achieved stable orbit and set its course for deep space. The initial phase of the voyage was dedicated to stabilization and calibration. Automated systems performed comprehensive checks on every aspect of the habitat, from the regenerative capabilities of its living walls to the metabolic performance of the synthetic symbionts thriving within. Meanwhile, a carefully selected crew of experts in biology, engineering, and medicine conducted daily rounds, ensuring that every component of the living system operated within its prescribed parameters. The departure from Earth was as much an emotional journey as it was a physical one. Families, scientists, and engineers alike felt the weight of history upon their shoulders, each acutely aware that they were witnessing the birth of a new era. The vessel's gentle propulsion, the soft glow of bioluminescent corridors, and the rhythmic hum of living systems combined to create an ambiance of quiet determination and wonder. As the Genesis habitat charted a course into the uncharted vastness of space, it carried the collective hopes and dreams of a civilization ready to embrace a future where synthetic biology would lead the way into the cosmic unknown. Aboard the Genesis habitat, the convergence of human ingenuity with synthetic biology was evident in every facet of daily life. The vessel's design provided not only a self-sustaining ecosystem, but also an array of advanced biotechnologies that enriched the human experience. Throughout the habitat, bioengineered interfaces allowed crew members to interact directly with living systems. These interfaces, crafted from responsive organic materials, adapted to touch, temperature, and even emotional cues, creating an environment that was as intuitive as it was futuristic. Within the medical bays, lab-grown organs and synthetic tissues were produced on demand by cellular printers employing the latest advances in regenerative medicine. This capability revolutionized healthcare aboard the Genesis habitat, enabling rapid recovery from injuries and seamless replacement of damaged biological components. The integration of synthetic biology into human physiology, extended even to everyday life food production, was managed by genetically optimized plants and microbial cultures that provided a perfectly balanced diet, all regulated by an AI that dynamically adjusted growth conditions to meet the nutritional needs of the crew. Educational and communication systems on board were transformed by the marriage of biology and technology. Holographic displays generated by bioorganic processors brought lessons on genetics, evolution, and interstellar exploration vividly to life, inspiring the next generation of scientists and engineers. In communal spaces, digital murals composed of living pigments shifted in color and pattern in response to the moods and rhythms of the inhabitants, a dynamic art form celebrating the fusion of nature and technology. The boundaries between human and synthetic began to blur as each individual's daily routine intertwined with the vessel's living infrastructure. Crew members were not merely passengers but active participants in a grand experiment redefining what it meant to live in space. Social structures aboard the Genesis habitat evolved in innovative ways. The community was organized around collaborative research and shared stewardship of the living environment. Regular forums and symposia enabled inhabitants to exchange ideas on further integrating synthetic biology into their daily lives, whether by developing new modes of biocivic engagement or by exploring genetic enhancements to better adapt to space. The relationship between humans and engineered organisms was mutually beneficial, while the crew maintained the health of the biosphere. The synthetic life forms provided vital support for human survival. This harmonious integration of life and technology was not just a necessity for space travel, it was a bold statement of humanity's evolutionary leap toward a future defined by the seamless union of biology and machine. As the Genesis habitat ventured deeper into interstellar space, the dynamic interplay between its synthetic systems and the natural forces of evolution began to reveal remarkable phenomena. Within the closed-loop ecosystem, engineered organisms were not static creations but living entities subject to mutation, adaptation, and natural selection, just as on Earth. Scientists on board meticulously tracked genetic drifts and phenotypic adaptations among the microbial consortia, engineered plants, and biofabricated structures that composed the vessel. One of the most intriguing discoveries was the emergence of a novel strain of luminescent algae that began to display patterned glows resembling a living constellation. These bioluminescent displays were not random, they appeared to follow subtle cycles that corresponded with the spacecraft's acceleration and deceleration phases. Researchers hypothesized that the algae were responding to the physical dynamics of the vessel, and that this adaptive behavior might be harnessed as a natural sensor system, capable of providing early warning signs of structural stress or environmental fluctuations. Early experiments with these luminous organisms yielded promising data, suggesting that they could become an integral part of the vessel's comprehensive monitoring network. Engineered microbes designed for waste recycling began to evolve enhanced metabolic pathways, increasing their efficiency in converting organic waste into vital nutrients. Driven by the unique conditions of space, microgravity, heightened radiation, and altered atmospheric pressures, These organisms adapted in ways that both surprised and delighted the onboard research teams. Their spontaneous evolution fostered new symbiotic relationships between the microbial populations and the human occupants. Each day on board became an unfolding experiment where evolution was played out in real time, offering insights that challenged established biological paradigms. Yet the evolution of synthetic organisms also raised challenging questions. While most adaptive changes proved beneficial, isolated instances of unexpected mutations led to overgrowth or imbalances that threatened the delicate equilibrium of the closed ecosystem. In such cases, the onboard AI, working hand in hand with human experts, swiftly implemented corrective measures, ranging from targeted biocontrol interventions to adjustments in nutrient delivery. These episodes underscored the inherent unpredictability of life, even when it was engineered, and highlighted the necessity of constant vigilance in managing a living spacecraft. The ongoing cycle of observation, adaptation, and intervention within the Genesis habitat provided a vivid demonstration that synthetic biology was not a static discipline but a dynamic, evolving field. Every discovery added layers of complexity to the mission and reinforced the belief that the rise of synthetic biology would ultimately be the key to unlocking humanity's future among the stars. No pioneering endeavor comes without its challenges, and the Genesis habitat faced obstacles that tested both its synthetic components and the ingenuity of its human caretakers. One significant challenge was the gradual accumulation of genetic mutations within the bioengineered cells forming the spacecraft's structure. While many mutations were benign, or even beneficial, some produced minor instabilities in the self-repair mechanisms. In response, the onboard bioengineering team, in collaboration with the vessel's AI, instituted rigorous monitoring protocols to detect and correct deviations before they escalated into critical issues. High-energy cosmic radiation also posed a constant threat. Despite the resilience of the living hull, sporadic bursts of cosmic rays penetrated its defenses, causing localized disruptions in cellular function. To combat this, scientists developed specialized bioshields, layers of genetically engineered radiation-resistant cells that could be activated in zones deemed high risk. Seamlessly integrated into the existing structure, these bioshields acted like an immune system for the spacecraft, rapidly identifying and repairing radiation-induced damage. The interplay between these protective layers and the habitat's natural regenerative processes underscored the synergy between synthetic biology and adaptive engineering. Management of microbial populations presented another continuous challenge. The delicate balance of the closed-loop ecosystem was susceptible to fluctuations in nutrient availability, temperature, and environmental pH. Occasional outbreaks of microbial overgrowth, or sudden declines in critical populations, required swift, targeted intervention. To address these issues, the research team implemented adaptive control systems employing real-time feedback loops. These systems could adjust nutrient flows, light exposure, and even local pH levels to ensure that no single species overwhelmed the ecosystem or diminished essential functions. Human factors added further layers of complexity. The psychological and physiological stresses of residing in a synthetic, ever-evolving habitat demanded constant adaptation from the crew. Comprehensive training programs and robust support systems helped inhabitants manage isolation, adapt to the gentle shifts in artificial gravity produced by the vessel's rotating biostructures, and maintain overall well-being. The integration of synthetic biology into every aspect of life aboard the Genesis habitat required not only technological acumen, but also a deep understanding of the human condition. As crew members worked hand in hand with living machines, they became both stewards and beneficiaries of a system whose unpredictability was matched only by its innovation. Every challenge encountered was met with scientific rigor, creative problem-solving, and an unyielding belief in the promise of synthetic biology. Through a relentless process of observation, analysis, and iterative adaptation, the Genesis habitat evolved into a vessel that was not merely a machine, but a living testament to humanity's capacity to innovate in the face of adversity. The lessons learned on board were destined to serve as the blueprint for future generations of self-sustaining adaptive living systems in space. After years of continuous evolution and refinement, the Genesis habitat began its final approach toward its designated destination, a promising exoplanet orbiting a distant star system identified as a potential cradle for human colonization. As the vessel's trajectory was fine-tuned to account for gravitational influences and the subtleties of interstellar navigation, the onboard synthetic systems entered a new phase of operation. The engineered organisms, honed by countless cycles of adaptation, now functioned with unprecedented efficiency and integration, a living mosaic of human ingenuity and nature's resilience. In preparation for arrival, the onboard teams redoubled their efforts to calibrate the habitat's adaptive systems to meet the anticipated conditions on the new world. Engineers and biologists collaborated to simulate the exoplanet's atmospheric composition, gravitational forces and radiation environment, making fine adjustments to the synthetic ecosystems accordingly. Specialized modules within the habitat were reconfigured as transitional zones, biological bridges that would interface between the living spacecraft and the alien environment beyond. These zones were populated with arrays of bioengineered plants and microbial cultures designed to initiate the terraforming process, beginning the conversion of the exoplanet's atmosphere into one capable of sustaining human life. The final leg of the journey was marked by a series of meticulously orchestrated maneuvers. The propulsion system, which had provided gentle yet steady acceleration for years, now initiated a carefully planned deceleration sequence designed to place the Genesis habitat into a stable orbit around its new home. As the vessel gradually slowed, sensors recorded the interplay between its living components and the external forces of deep space, gathering invaluable data for future missions. In a moment of quiet triumph, the onboard AI announced that the vessel had successfully entered its target orbital path, a milestone that signified not an end, but the beginning of a bold new chapter in humanity's expansion into the cosmos. Throughout these final stages, the crew maintained a sense of cautious optimism. They had witnessed firsthand the transformative power of synthetic biology, and now, as they prepared to transition from their living vessel to an alien landscape, they looked forward to the challenge of establishing the first footholds on a new world. The Genesis habitat, with its ever-evolving network of engineered organisms and adaptive systems, stood as a beacon of hope. It was a living, breathing promise that life, in all its synthetic glory, could overcome even the most daunting challenges of space travel. The arrival at this new world was not simply a destination reached, it was a testament to the resilience of the human spirit and the infinite possibilities unlocked by the fusion of biology and technology.